So I think we can move on to we're going to move on to the next session on our agenda today, which is a statistical software session for imaging analysis. So let me briefly introduce the background of this session, and then I'll pass the torch to the session moderator, Shijia. So the software session is another new session that we launched at SMI 2021 this year. And the objective of this new session is to provide a forum for our imaging statisticians to showcase the platform or software they have developed for disseminating advanced statistical methods for imaging analysis. For those of us who work in imaging, we all know how important it is to have user-friendly and also reliable software packages. It has been increasingly and crucial to kind of promote the impact of statistical methods in the imaging community. So our hope is that this kind of an imaging software session will help encourage and promote software development culture among our community. So today we have 5 speakers here to demonstrate the platform and the software they have developed. And we hope in the future SMI conferences and also other statistical conferences, we'll be able to see more researchers and team to demonstrate and share their software packages. Okay, so Shijia, you can start the session now.
So hello everyone, welcome to session 3 of Statistical Software for Image Analysis. My name is Shijia Bian. I'm a second year PhD student at the Department of Biostatistics and Bioinformatics at Emory University School of Public Health. And I will be the chair for today's session. And our session will start at 2:55 and end at approximately 4:35. Each of the speakers will have 20 minutes to present. So if we have time, at the end of each talk, we can have the speaker to answer questions from our audience right after their talk. You can type your questions to the chat box. Meanwhile, we will have a 20 minutes floor discussion right after our session, which will start at 4:35 and end at 4:55. All audiences are welcome to remain in this same room to continue our floor discussion. So without further ado, let's introduce our first speaker, Dr. John Muschelli. So Dr. John Muschelli is currently an associate scientist from the Department of Biostatistics at Johns Hopkins Bloomberg School of Public Health. His work focuses on processing and analyzing medical images, especially brain image. Dr. Muschelli is also a very active contributor to the art community. His talk today is titled Neuroconductor, an art platform for medical image analysis. Let's welcome him.
Thank you. Thank you for the introduction, and thanks to the committee. I know it's been a year, but I appreciate all the work you've done to organize this. Sorry we couldn't be together, but let me get started. So I sent out a link to my slides, so that should be on my GitHub somewhere as well. And if you check, I was doing some updates, yesterday and today, for example. So even if you give at least me a year, it doesn't matter. I'm still going to do slides. The week of, I'm just telling you. So I'm going to be talking about Neuroconductor, which I've talked about a few times in this endeavor, and some overall things. So I'm going to start out with some little hot takes. So biostatisticians or statisticians, we were late to the game, the neuroimaging game. Engineers kind of paved the way really early, but I do want to at least give a lot of respect to the founders, as Tim is definitely one of them, who really paved the way for statistics and imaging, specifically neuroimaging, to kind of get up and running. A lot of people who are on this call, I'm not going to name everybody. I think you know who you are. So engineers really got there first, right? A lot of MRI physicists also as well, MRI physicists, right? They had to do a lot of the engineering with respect to the magnets and getting everything going on. And MRI is a huge player in the field, right? And because a lot of the stuff came out of engineering, Matlab kind of reigned supreme, right? So if you wanted to do anything and everything, you pretty much looked at a Matlab toolbox, and things have changed over the years, right? So SPM is still a fantastic MRI-based software to do things, but I think now Python is really stepping in a lot of those gaps because it is a very wide scripting language. NiPy is a fantastic suite of tools for imaging, especially neuroimaging. I will be talking more about medical imaging in general, but I'll be focusing a lot on neuroimaging because that is still where a lot of the imaging game is. Also, I will say, some of the Python community, they have some fantastic logos as well. I mean, the fact that this is a Python wrapped up into a brain, I mean, it's hard to beat some of these things. So I'm just saying their branding is on point. Now, I'm not going to be talking about Python or anything like that, but I am going to be talking about some general things in R. So if you've never seen the movie Margin Call, it is a good movie, well acted. There are some people who've done some probably not so great things in their outside of life, which I'm not going to touch on, but there is a very specific line in there that I think about a lot, and it's from Jeremy Irons. It says, there are 3 ways to make a living in this business. They're on Wall Street. Be first, be smarter, or cheat. And he says, and I don't cheat. I'm not going to say that because I do cheat. So I say you should be first, or be different. Sorry if getting any of that. Also, you should steal things. Steal things from other people who've already done really great work. I think I remember in grad school, I heard someone say, if a new piece of data comes out, or a new image or data type comes out, and you're the first to do the SVD on it, you will do really well in that field, right? So the first to SVD in some respects in some new data types. And I'll talk about what I mean by stealing explicitly. Sorry, this isn't moving. Oh, so as many of you know, what is R? R is a language and environment for statistical computing and graphics. And I will highlight the statistical because that's how it started. But what it should say is R started as that. So I've been working in imaging in R for about almost over 12 years now. And when we started, Shiny didn't really exist. Like Plotly wasn't a thing. ggplot2 wasn't out there. dplyr wasn't out there. Like a lot of different things. You can do a whole bunch of stuff with R now. Plumber wasn't there to spool up APIs. So R started as a statistical language and grew more as a general purpose tool, whereas Python started as a general purpose tool and has narrowed in some of the statistical frames. And MATLAB's its own thing. MATLAB's doing math calculations in many respects. So this is me in grad school. A couple of years ago, grad school was really rough. So I had some more wrinkles. So this is what it looked like, I think, a few years ago. So what did R have for medical imaging? So if you looked around, it was hard to find them. And this is what we have. So we have a medical imaging task view, which is maintained by Brandon Witcher, who's a contributor of Neuroconductor. I use a lot of his packages like Oro.nifty and some of the other ones if you know R. But if you're a new user starting out, this is not... just say that not the most intuitive. It's a very, very old looking website. That's because it is. It's just a website with links, but it's a great starting point, but it's not the most intuitive thing going forward. So starting at things like this, we wanted to try to make things more intuitive for the new users coming out. So this is the envy I have. It's deep in my soul as someone who does imaging in statistics. It's Bioconductor. Bioconductor is a centralized bioinformatics platform. It has over like 13, maybe even 1,500 packages now. Great community, huge development team. They have workflows and tutorials, and they're like a high caliber code base, right? They do some great stuff. They have additional requirements to CRAN. CRAN is the huge repository of packages for R. And when I was saying back here, I was saying, where is the imaging R packages? That has gotten even worse now in some respects because CRAN has over 15,000 packages. So navigating that space is not easy. So Bioconductor is kind of a one-stop shop. You want to do bioinformatics in R? That's where you go, almost like period. So this is the types of things. A few years ago, it's probably about six years ago now that we were like, why can't we have Bioconductor but for imaging? And here's the question of why you should care about Bioconductor. Why should these things matter? So I have heard many students over time where I'm like, why don't you learn imaging? Imaging's super cool. And they say, I can learn genomics with Bioconductor a lot easier than I can do for imaging. So recruitment of students sometimes is inhibited. So researchers see analyses. So in Bioconductor, they look at a package or a workflow and they're like, that's the exact thing I want to do. I just need to put my data in there. It gets the bar way lower to actually doing a full-blown analysis. And the truth is, papers with software get cited more. I don't have a citation for that. Maybe it's a bad indication, but they usually do. But also many times, we can discuss the merits or the pros and cons of this, but many times there is a paper that comes out demonstrating an analysis or a new method. And then there's a completely separate paper that talks about the implementation in the software. So I'm not saying you should try to quote unquote double dip, but usually there are two very distinct processes for research. And usually you can publish on both of them with not having exactly 2x the amount of work. So all of these things usually lead to funding, being able to recruit students, do analyses easily, and get cited. So I will say this. I think there's enough space for Python and R and Matlab, but statisticians, we need our niches or niches, however you like to say it. And I think some of them are here. So this is what I'm saying, medical imaging. We called it Neuroconductor because it sounded good. And this was 6 years ago. And once you name a thing, you can't unname a thing. So we call it Neuroconductor, but it should be like medical conductor or medical image conductor, but that doesn't really roll off the tongue either. So Neuroconductor it is. But there are, your body's not just your head. So a lot of places the body are imaged. The tools for that is actually way fewer and far between compared to the head. So if you're trying to say, I want to make my mark somewhere, focus on a part of the body that isn't the brain. That's my general take. So also MRI is almost magic. You go into a machine, they shoot magnets at you. That's not how magnets work, I know, but they shoot magnets at you and you get a picture back of inside your body. It is borderline magic, but there are other imaging modalities out there. FNIRS, CT, PET, EEG. And again, this is where I think a lot of the statistical prowess we provide can make a huge impact. Also, mixed effects and longitudinal modeling. That is our bread and butter. And that is becoming a lot more common in the types of data sets going out there now. So it's not just, we had a study, we brought 20 to 100 people in, we scanned them, we did the analysis. It's now we did that, but we have monthly intervals, we have biobanks coming out with longitudinal data. And this is kind of a fertile ground for statisticians, I think, to make a large impact. And in that vein, data harmonization is huge. So genomics have battled this problem from the beginning, and neuroimaging in a lot of respects, they've had issues with it, but it's becoming a lot more problematic when you have multi-site data, data from all over the country, or you have scanner changes, these types of things. So this is what I'm saying, steal. So genomics have done a lot of time, have put a lot of time and energy into data harmonization, and we should try to adapt those methods if they are appropriate to imaging data. Some of them are not, some of them are, which I'll show you in a minute. And the other thing we usually have as a really good niche is Bayesian just machinery, even though it's very, very problematic at times for the size of the data we're talking about. But that is where we have a holdover over a lot of engineering and a lot of other bioinformatics groups. So label fusion, statistical properties of things like that, because when you do like label fusion, you're doing a bunch of registrations where you kind of have an estimate for each target to a template, and then you average them or something like that. And statisticians are super good at taking a bunch of estimates, averaging them together, and getting statistical properties from that. That is like, I mean, if you were to ask me really hard what our field is, that's pretty close. So these are 3 papers from an old colleague and an old Hopkins alum, Jean-Philippe Fortin. And what he did was, he had DTI data, he had MRI data, things like that. And he said, hey, I have a bunch of data from a bunch of other scanners. I do genomics. Let me take some of the genomic stuff and apply it to it. And I'm not using citations as like the best metric of a paper, but these are highly cited because he took kind of almost off the shelf genomics, harmonization procedures, and applied it to imaging data. And I think this area is growing very strongly. I know a lot of people in the audience are doing things like this. So I think this is where we can make a huge impact with the data changes and the longitudinal aspect of data coming up. So this is why we tried to get Neuroconductor together, right, in our platform for medical imaging analysis. So we have 115 packages. We have a community of developers, so it's about 30. So there still isn't as many, but if you have a package, we would love to submit it. So I'm also really excited. There is some great pieces of software getting talked about in the rest of this session. rtemis I've looked at is a fantastic comprehensive system. That is a thing almost on to itself. So Stathis is gonna, go into detail about that. But the other things, we've tried to provide some tutorials to say like, hey, you want to learn how to read an image, you want to learn how to do like skull stripping, these types of things. I believe these are the training materials we need to provide to students to say like, hey, it's easy to do imaging too, so that they don't just have this easy off the shelf answer to say like, I don't need to, imaging's hard, bioconductor has all these easy things, so we can at least lower the bar for them. So we also try to help developers and users like learn, so making these tutorials, but also getting developers on our system quickly. And we are focused somewhat not just on analysis, right? So data processing, so getting some from raw images into something you can actually work with is non-trivial, and in like 9,000 different ways it can affect your end results. So knowing kind of what happens from knowing what happens to your data from like soup to nuts, I think is very important, and we can provide a system that allows you to do that. So also, like, you have R, right? There's a bunch of other stuff that's going on now that are super cool. So we saw today machine learning, so TensorFlow packages, Keras packages, versioning and testing. So GitHub can provide us like how to keep code stable, reproducible reporting, R markdown documents, like interactive documents you can upload with a click. Like it's, R is just pretty good. I'm a big evangelist of it, but it has a lot of other capabilities other than just imaging. And it's great when we can leverage it too. And I think the last bullet point is really the big one, where you have genomics analyses and imaging analyses. You can at least combine them in some way. I'm not saying, we exacerbate the type 1 or the type 1 error rate problems in both of them and make it doubly hard. But sometimes you have genomics data, you have imaging data, and to be able to do that in one single platform is really helpful. So what do we have kind of going in there now? So you want to do image registration. So there's packages to do it. ANTS, so Advanced Normalization Tools from Brian Avant's group. Our Nifty Reg from John Clayton and the Nifty Reg kind of backend, FSL and FSLR, to be able to do like image registration. Getting things in the same space is definitely possible. So let me check. Is mixed effects modeling being used on unstructured raw images nowadays? I don't know. I'll pick that up maybe in a little bit after I go through this. So also almost every single thing, if you have structural imaging, you're going to do inhomogeneity corrections. So you have them in ANSR and FSLR. You want to make sure that this area of the brain and this area of the brain, if they kind of have the same properties, give you somewhat of the same answer with respect to intensity. Intensity normalization. So Takeshi Nohara and his group have done some really great things. So White Stripe is one of those examples that tries to normalize the intensity of the units in MRI to normal appearing white matter. Ravel and Combat are two, again, processes that were taken from the genomics literature and applied to imaging data to try to make it more harmonized. Image smoothing, so FSL, ANSR, Analyze fMRI have a number of different 3D Gaussian smoothers and some anisotropic smoothers you can apply to your data. So sometimes, if you can figure out how to smooth the data right, maybe your statistical model can be a little bit more simpler, or your segmentation can be a simpler thing, because going from something like this to something a little bit more smooth might allow you to get a lot of type 1 or type 2 errors kind of knocked down already, while also using spatial information, but not having to impose that on the model with a strong spatial prior or doing anything like that. You kind of just pre-smooth the data and kind of treat it like it wasn't smoothed. So also accessing data. So there's packages for connecting to the Human Connectome Project, some of the Kirby 21 data, which is 21 subjects that are scanned twice, which have T1s, T2s, fMRI, DTI, a whole bunch of other things. So it's a good replication data set and example data set to do work on. NITRIC, which is a huge clearinghouse of neuroimaging data and tools. RxNat allows you to connect to XNAT databases, which was a lot of people stand up for their imaging work. Template Flow, there's a package for that to connect to templates. So there's like an API where you're like, I need this template. So like an MNI 152 nonlinear thing, you can grab that programmatically versus having to download that kind of on the fly. And then you forget 6 months later, where did I get that from? So you go to write the paper and you're like, what template did we actually register to? I can't be that specific because I don't know, but if it's in code, you're good. So also check out some of the tutorials. There's a link to the Neuroconductor website. It's also on my website here. So on showing you how to do some of the tasks that I find pretty useful in imaging. So segmenting a brain, skull stripping a brain, things like that. And then we have actual more formalized courses. So we have one on Coursera called Neurohacking in R. The imaging in R is on my website, which is a little bit more, it's kind of a neurohacking like 1.5. I wouldn't call it like a 2.0. Doesn't really replace it, but it adds some things on there. And then we have some other links there to Martin Lindquist's fMRI courses on Coursera, which are really good. The other thing, especially because we've spoken about about deep learning tutorial today, there's a thing called AntsXNet, which is a whole plethora of models that are already fit for a specific purpose. So brain segmentation based on patches, proton MRI lung segmentation, functional lung segmentation. You know, I don't have any pig lungs that I've imaged, but if you do, then pig lung segmentation, brain age estimation from structural or functional imaging, things like that. They have a lot of deep learning networks out of the box, or that have already been fit that you can kind of use almost out of the box. We had one that we made a convolutional neural network for stroke segmentation. There's some tools for EEG and some other platforms like that. So wanted to make sure we're on time. So I flew through a lot of that stuff. So let me jump into the chat, but open up your mics and let me know if you have any questions.
Let's welcome our second presenter. So our second speaker will be Dr. Stathis Gennatas, and he is currently the assistant professor at UC San Francisco, Department of Epidemiology and Biostatistics. He is also a member of Center for Intelligence Imaging. His research is focused on machine learning for precision medicine and neuroscience. The topic he will give us today is titled Efficient and Accessible Machine Learning with Archivists. So let's welcome him.
Thank you very much. Thank you. And thanks everyone for organizing. It's great to be here. Of course, I would be happier if we were meeting in person. I'm sure a lot of you would agree, but it's still very fortunate we can do this. Let me know if I need to adjust my volume up or down. So I'll go straight to it. And I'll give you, first of all, that was a great talk, John. And I have to attest, I use a lot of that software, and it's not just a lot, it's very well written, very high quality. I was writing a new AnsarNet pipeline just last week that worked beautifully. So I'm going to give you a quick overview of this machine learning package that I've been working on. So let me give you a little bit of a background. And here we go. This is Mission Bay campus from just before the lockdown. Here we go. So what is this? We're talking about a machine learning package, machine learning and visualization package, which is targeted at everyone, regardless of experience and expertise, level of expertise, whether you're training a model, predictive model for the first time ever, or you're a total expert and you've written a dozen of your own algorithms. And the goal is to maximize efficiency. It's pretty straightforward, but of course, never at the sacrifice of control of the whole pipeline, of all of the parameters, of all the steps, just to make it as painless as possible, essentially to remove the technical hurdle from the process, whether you're working on basic biomedical research or clinical research, whether you're an expert in statistics or not, or in basic science or clinical medicine. You never want to not waste, but spend more time than you need on what is essentially truly technical and can be automated. So if it can be automated, it should. If it can't, it shouldn't. This is the logo. This is driving from Marin County to San Francisco. It's a lot of symbolism that I think fits with the machine learning story here. So the motivation, how did this whole thing start? My background is I went from medical school to full-time research in neuroscience, neuroimaging, and PhD in brain development. During that time, I had a big data set, about a couple thousand cases and a few thousand regions of interest. And I wanted to do a couple of things. I wanted to tune sparse decompositions to reduce the dimensionality and tune grain boosting models to predict different outcomes of interest. So that was when I first started coding in R, and I started with these two functions, and then, a little bit later it became a few hundred functions. And this is what I want to show about today. This is just a very sort of non-comprehensive idea of what's included in this software package. So it's written in R. It has a lot of functions for graphics, split into sort of static and interactive or dynamic graphics. So a lot of plotting and different types of graphs. Unsupervised learning, so clustering, decomposition, supervised learning, classification, regression, cross decomposition, meta modeling, where you have models whose input is the output of other models. And I'll talk more about the structure, very, very briefly about the technical implementations for those that are might be interested. So the principles in general, again, we want to minimize user time. That's sort of obvious why. So we have flexible high-level functions, but we're not limited to high-level functions. It's by no means a full auto. It could be, but you can work at any level. You can use the lower-level functions, the higher-level functions, and so on. So you have full control to experiment and so on. But at the end of the day, you can collapse everything to a single call per model, as I'll show an example of later. Again, we do our best to minimize machine time by parallelizing on or using distributed computation. We try to minimize user error. I forgot to start my timer. Minimize user error by minimizing the amount of times you copy paste. Everyone's kind of work requirements are different. It's certainly common that you have to train thousands of models, maybe in a single evening. So as much as you minimize the opportunity for error, of course, it will pay off. We want it to be user-friendly and intuitive. Again, pretty obvious. Try to minimize the need to consult the manual, and when you do, try to make it as easy as possible. And make the whole experience as informative. So there's a lot of messages that tell you who is doing what, meaning what function is doing what, try to have very informative errors and so on. And it's very modular so that you can actually use it to build all sorts of new pipelines and procedures. And it's a very large package because all the different parts are interconnected. So it wouldn't make sense to split, for example, the visualization functionality out of the rest, because the supervised learning will use the plotting functions. The plotting functions will use the supervised learning to do fits and so on. And it comes together pretty nicely. In general, we use it for research. So I won't talk about this today, of course, but there's a number of algorithms that I've worked on with this big group of collaborators. So they've been built directly on rtemis, which makes it easy, again, because of that infrastructure, that modular infrastructure. So it makes it much easier to make the final, both testing and sort of the final packaged algorithms. We use it in class. I teach a couple of health data science class at UCSF, brand new curriculum. Shout out to John Cornack. And for applied data science, I've been using it nonstop for many years now for all my projects, with a lot of collaborations. Again, trying to make it as efficient as possible. And when you have such a nice, tidy, compact code, it's much easier to share. It's much easier for someone to understand, to reproduce, and so on. This is an example of what it looks like. A simple model on everyone's favorite data set, I guess. A lot of time has gone into the design and the color matching between the console and the plots. I'll show you more of that. Again, going through a number of things briefly here to give you an idea. Visualization, a lot of functions, all sort of more or less obvious: univariate, bivariate, multivariate plots, plus a few other things. A lot of decision trees built in because that's part of the research I do with collaborators. I put in a bunch of mapping functionalities during the pandemic because of research on that. So, of course, kind of everything that's in here is because I use it more or less. Again, more examples, 3D plots. So the mplot3 functions are the static graphs based on the base graphics, and dplot3 are the dynamic, most of them based on Plotly and other interactive libraries. Very briefly, the advantage of the static graphics of the base, specifically base graphics, base functions are very fast. They're easy for users to extend, and they're composable. I only have one grid graphics-based function to make pretty maps, and the interactive, which work great with either Shiny apps or are Markdown. And for some reason, again, there's a lot of attention to design, which is sort of just for fun mostly. And there's more than 50 universities' color palettes. If yours is not in here, I will add it. You just have to email me. So this is just an example of the theming. So this would be the default theme, and you can set system-wide defaults. And I'll go to sort of the main part. I guess it's the main interest because it's the more involved. It has the most demanding sort of code. And looking through a supervised learning pipeline, there's all the different parts that you might need to use. So first, after you're reading your data, there's a check data command. We'll look at an example output that will give you sort of diagnostics and will print out in plain English what it suggests that you do. For example, if it finds that there's duplicated cases or constant variables or missing cases and so on. The visualization functions, again, my sort of statistics teaching started with breathe, visualize, model. So after you breathe, you visualize, I suppose. And I do believe in that. There's a preprocess function, straightforward, will do all sorts of operations for you. Any type transformations and other encodings that you might need. Pretty straightforward names. Again, that's the point. Resample, different types of resampling. And then you have two choices. You either will train a single model, so you have a whole list of names, about 80 different algorithms, which helps because then you can auto-complete to find all of their parameters. And that includes automatic tuning, which I will show you. So all you have to do is you give, instead of a single value for a parameter, if you give a vector with 2 or more values, it will automatically induce a grid search and find the best combination, retrain on the full training set, and give you the training and testing metrics. And there's a step further. If you want to do this nested for multiple resamples, then there's another command that I will show you an example of. And finally, there's another set of visualization functions for plotting the model performance and decision trees and survival plots and all sorts of beautiful things. This is an example of, it might be a little small, of the check data. It gives you a summary. And again, it tries to be sort of pretty straightforward. There's color coding. I think if it's something really bad, it's going to be red. By I think, I mean, I know it is red, but it's always trying to be as helpful as possible. So the idea is you run this, and then based on what you get here, you feed that into pre-process. So let's go into an example of how you would do a pretty simple, straightforward training of a model. So on the left, we'll have simple R code, on the right, the description. So you might start by reading in a CSV, and then you have your data right there. The easiest thing you would do is just zero effort. You throw that into the elevate function. That was a code name, but it's stuck. It's the one that does the automatic nested resampling. It sort of does everything for you. So by default, this will give you a 10-fold cross-validated random forest for classification or regression. And that's already pretty good, especially if you have no experience in modeling. It's not the best, but it's certainly really great. And suppose let's say you want to do better. You want to use gradient boosting. All you have to do is specify the model. So you say mod, give me GBM. So now I will do 10-fold cross-validated gradient boosting. And then suppose you want to do hyperparameter tuning. You say, well, I want to try these values. Like I said, you just give a vector of values for, this is interaction depth, the depth of each base learner, of each base 3. And the shrinkage, that's the learning rate. So it will do a 3 by 2 by 5, which is the default cross-validation number of resamples. And then suppose you also want to do pre-processing. I generally would do it separately ahead of time, but of course you could, again, collapse it all into a single function. You can set, let's say you have missing data, you say impute is true, and that will do the, by default, there's a number of methods. The default is the iterative random forest. So suppose then you have a very high dimensional input and you want to also do a decomposition before you begin. Then you will say decompose. Let's say I want 20 ICA dimensions. And then let me see if I should be checking. I'll answer the chat questions at the end. Let me see. So then if you want to do, you have control over the resampling, you can say I want 25 stratified subsamples. Suppose you have a smaller data set and tenfold is not good enough. And then you can save everything that happens into an output directory where it will save the model in a binary R format. It will save the plots, the performance for the training and the testing, and so on. This is an example of the output. Very briefly, it's for those that know about R class systems, it's an R6 object, but what that means is that it allows you to save both data values and functions within an object. So this will be your model of whatever you are looking at, and it will hold both the different resample training and testing errors, the parameters used, plus the functions to create all sorts of plots. In this case, it was a classification. So you have your confusion matrix, you have your ROC, variable importance, and there's a number more, a lot of options here. And the good thing is that everything's self-contained. You don't have to wonder, okay, I have an object. Of course, there's ways in R to find exactly how many different functions you can apply in that object, but most people wouldn't necessarily know how to do that. And this is, again, in keeping everything tidy and accessible. This is just another example. If you were training gradient boosting to get an idea of efficiency, essentially, this will do automatic hyperparameter tuning. In this case, 3 by 2 by 2 by 5 plus 1. So the grid plus the final retraining on the full training set, 61 models. If you were then to do the nested resampling, this single simple call would be times 20, so 1,220 models. And imagine I have a set of experiments, and what we usually do is have an outcome of interest, and of course, no free lunch. You don't know ahead of time which one will be the best algorithm, but you may want to try your regularized linear model, your card random forest gradient boosting plus something fancier. And essentially, each one of those will be one call. And at the end, you just make a nice box plot of their performance and say, hey, here we go. And you can go on from there. Now, this is an example of sort of the most popular, at this point getting outdated, but still most popular sort of equivalent package in R, which was Caret. So that's what I first looked at and never used for a number of reasons. This is taken from their documentation. Again, I'm not trying to say anything against Caret. I'm just saying why I built a different package. And this is for the example from their documentation about essentially building a gradient boosting model. And the exact same code is here, but not really exact, because this is only doing the inner resample, so it does the tuning, but then you have to retrain separately, get the error metrics separately, plot, and so on. And one of the problems with Caret is it never suggested that you should. Of course, you could if you knew that you should retrain on the full training set, which can be very important, especially on a small medical data set. And another issue here was that they don't do proper tuning of gradient boosting, and instead of training to a max number of base learners, I suppose a number of you would be familiar with this, and then look at the validation curve. maybe smooth it and find where it's minimized. They cross-validated the number of trees, which is incredibly inefficient, incredibly slow. And of course, you're only going to try a very small number of discrete values for the number of trees, which is certainly not going to be optimal. And just because it was the very first thing that I wanted to tune, that's why I never used it. So at this point, I was going to do a short demo. And this is what it looks like when it starts. It has a random image, and I'm going to load this simple Sonar data set. It's sort of a standard benchmark. Create a resample, a training set, a testing set, and then I'm going to do random forest with hyperparameter tuning. So for the M-time, the number of variables to consider for at each time that it's training, at each split, it will consider what is sort of the default, which is about a quarter, and the full number of variables. And I'll show you the output. It's already finished. Of course, it's a small data set. So starting here, I hope you can see it. I'll make it bigger. I don't know how many. Well, that doesn't. I have a small screen, so it gets crowded when I magnify it. But it says hello, and you always get the date, the time, and the name of the function that is outputting of whatever message. And that's important. So this is the one you called, so that's why it's the first one. And then you notice that it gets passed to data prepare. And the data prepare function found that it's imbalanced classes, so it applies IPW, inverse probability weighting. I'll speed up a little because I know I don't have much time. Classification input, parameters, resampling, it does the grid search. It says best parameters to maximize balanced accuracy using a full set. It's probably very close either way. Training was perfect, so it's like overfitting, but testing wasn't too bad. And you get your confusion matrix. So let me go back to this. I'll love to show more, but I will run out of time. A long list of algorithms, pretty standard. Hopefully everything you would need and a little more. Same for clustering. I'm going to go through them. Composition, linear, non-linear, even some autoencoders. Again, it's what a lot of people call a meta package. So of course, the point is to homogenize the input and output. Every package has different requirements. Is it a data frame input? Is it a matrix input? Is it a vector? Is it a factor? Is it a numeric 1, 0, and so on? So of course, it will take care of all of that and give you informative messages if there's anything wrong with your data, if the number of features don't match in training testing, or the number of cases or anything, or any problems. And it does parallel execution by default. We'll use as many cores as possible, which of course you can control. And there's a few options using essentially Spark and H2O if you have such servers available, where you can use the same rtemis interface to submit to a remote server. And then you get the results in the exact same format as any other model. That's the parallel execution. I'm going to go show you a brief overview. Extensive documentation and vignettes, self-explanatory. There's a very quick introduction. How do you do regression? How do you do classification in a single model or a cross-validated model, and so on? Gives you the output, and you can easily copy paste and follow along. And it gets more advanced. There's functionality to do bagging or boosting on any learner, which can be quite interesting. And you can learn a lot about bias variance trade-off. There's examples here. If your step size is too big, or learn rate or is too high maybe, and your trees are too deep, and so on. So there's a lot of material. Again, it should be very easy, sort of copy paste, try it on your own data. There's functionality to write your weights, whatever variable importance it is, from whatever model into nifty images. So again, my work is in neuroimaging, so that's what it would be. But if you have a labeled file, a nifty format, I guess that would be usually brain imaging. You can write out the weights directly to there. The thing that I might not be worth going too quickly through it, but I'll show you just as a teaser. Essentially, this is the live version of rtemis, which is not available yet. So I'm not sure when and how. It's brand new, but it puts all the rtemis or most of the rtemis functionality in a Shiny app that works automatically. Let me see what I was trying to do. I was not here, I was here. So I believe I need to rerun this because I turned off the browser. So if we simply give a data set to rtemis Live, it loads. Now this will be super fast, but you get an idea. You have all sorts of plotting available on the first tab. You have your standard data query operations. You want to filter, select, group by. So you identify your cases, your variables, and your grouping factors. And you can have box plots, histograms. You can separate them. You can have density, overlap. You can have scatter plots, 3D scatter plots. The most important functionality here is the dark mode, because everything looks better. And I work at night, so I'm sure a lot of you do too. I'm sure radiologists prefer dark mode and everything. There's a heat map, pretty straightforward. There's the sort of interactive version using Plotly, and the base version, so you can sort of, do all the nice things, zoom and get hover information. You can do decompositions. Again, you have a whole series of options available, and I have to reload this. Yeah, of course it would do this right now. I need to restart the session, and there's a graph functionality. Again, different libraries to plot. Essentially, you make networks from correlation matrices using any type of correlations, edge thresholds, layouts, and so on. Cross-tabulation, you have an interface for hypothesis testing. It will automatically do t-test, Wilcoxon, ANOVA, Korsakoff-Wallace, the Fisher test, and chi-square. And I will wrap it up because I know it's late. You can do pre-processing, you can make automatic table once. There's a help and stuff that I didn't get to show you. There's an interface to John's MR, the Papaya R library to load MR images. So let me finish up here. Everything has its own logo. That's Pacifica, rtemis Live. There's ports in Python, which is not developed because there doesn't make sense necessarily. There's a Julia one, which is pretty great and fast. This is San Clemente. And a number of algorithms we've developed on the platform. And to end, any feedback, any questions, specifically if there's any specific functionality you look for, you would like to see in an ML package, and what algorithms and or pipelines you would like to see implemented. There's some great feedback. I haven't advertised the package, but people have found it because it's been online for a little while. And I will thank you and leave it to that. I'm sorry that I went a little bit over.
So for the next, let's welcome our third speaker, Josh Lukemire. And he recently defended his PhD thesis in the Department of Biostatistics at Emory, and he is now starting a postdoc. His research focuses on statistical methods for studying brain networks. His talk today is titled Hint, a Matlab toolbox for hierarchical covariate-adjusted independent component analysis of fMRI data. So let's welcome him.
Thank you. So I'm Joshua Lukemire, and I'm going to be presenting a toolbox today that we call Hint. And so this is a toolbox for doing hierarchical covariate-adjusted ICA of fMRI data. And this is work with Amit Verma, Yikai Wang, and Dr. Ying Guo. So the focus of this work is on looking at group and individual effects on brain networks. So these can be things like clinical or demographic covariates, and trying to understand how they change the spatial network organization of the brain. And so one of the most commonly used tools to answer questions like this is going to be spatial independent component analysis. And generally what you'll see in the literature when the question of interest involves how clinical or demographic covariates affect the brain network, usually what will be done is some sort of multi-stage procedure. So something like dual regression. And so in one stage of this, there'd be a temporal concatenation group ICA, followed by some sort of back reconstruction to obtain subject level brain networks. And then these subject level brain networks can be regressed against whatever the covariates of interest are to obtain covariate effect estimates. But there's some downsides to using these two-stage procedures. So one downside of using two stages is that there's potentially a loss of accuracy and power when it comes to actually detecting those covariate effects. And then also, as an aside, most of the existing ICA methods are developed for cross-sectional studies. But of course, a lot of data that's being collected now is coming from longitudinal studies with repeated scans on subjects. And so we'd like to be able to accommodate that type of data. So the hint toolbox that I'm going to be demonstrating today is implementing methods from a hierarchical ICA framework. And so what the real advantage of this framework is that it gives us a way to directly incorporate covariate information in and between subject variation into the spatial ICA decomposition. And so there's a few papers out now showing that when you take this type of approach, you can obtain improved accuracy and statistical power over other two-stage approaches like dual regression when it comes to detecting group differences. And these advantages seem to be especially pronounced in smaller samples. So as an example here on the screen on the right-hand side, I have some results from a simulation study of longitudinal data. And so the red lines here are going to be a longitudinal hierarchical ICA approach, and the blue line is a standard dual regression approach. And so this top row here is showing the type 1 error analysis for a couple different hypotheses. And then the bottom row here is going to be a power analysis. And so the main takeaway here is just that the red line corresponding to the longitudinal hierarchical ICA approach has quite a bit better power than the dual regression approach while maintaining a type 1 error rate that's closer to the nominal level. So there's been a handful of methods developed under this framework so far, and I have a list of some of them on the screen here. Today I'm going to be focused on two of them. So the first one is going to be the hierarchical covariate-adjusted ICA method, and this is going to be a cross-sectional approach. And then the second one that we're going to focus on today is the longitudinal covariate-adjusted ICA approach, or LICA. And this is going to be a longitudinal extension of the standard HCICA approach. So to give you some idea of what the HCICA model looks like, so there's two main levels here. And so at the first level, the observed fMRI data for a given patient can be decomposed into a mixing matrix times a set of subject-specific independent components. So these are the subject-level brain networks that we're interested in, plus some subject-specific noise term. And then what HCICA does is it takes these subject-specific brain networks, spatial components, and it models them in a second level as a linear combination of a population-level signal, or S0 map. So this is kind of like the brain network as it is common across all subjects in the population, plus some covariate effects. So these would be representing ways that things like clinical or demographic covariates are changing the brain networks that we're interested in. And then finally, there's a subject-specific random variation. So by taking a two-stage hierarchical model like this, we're able to directly incorporate these covariates into the ICA decomposition. And then we can carry out estimation using an EM algorithm. For the longitudinal ICA approach, the general setup is pretty similar, but now instead of modeling a single set of networks for each subject, we instead have a set of networks for each subject at each visit. And then we model those networks in the second level as a combination of, again, a baseline map or S0 map, but now we have visit-specific estimates for the covariate effects. And then we also include a subject-specific random intercept, which lets us pull some information across the different visits. So the toolbox I'm going to be demoing today is called HINT. And so this is a user-friendly toolbox for doing analyses under this HCICA framework. And so some of the features of this toolbox are going to be that it lets us specify different brain network models. So we can include different types of covariates, like continuous or categorical covariates, as well as interactions between them. We can carry out estimation for the cross-sectional and the longitudinal models using an EM algorithm. And then we've created pretty extensive visualization tools for examining those independent components and doing hypothesis testing for the results. So this toolbox is available from this GitHub link here. It's also currently available on Nitric. So right now what's available is the first release, which has fully implemented the HCICA approach for cross-sectional data. But we are also almost finished with a longitudinal ICA implementation, which will be in a second release of the toolbox, which should be coming out pretty soon. And in fact, that's what I'm going to be demoing today. So for today's demo data, we just generated some longitudinal simulation data for illustration purposes. We used 3 components in this data based on some brain network maps that were identified using temporal concatenation group ICA. So one of those is going to be the default mode, one is a sensory motor network, and one is a frontal network. And so I've got the default mode example on the screen here now. So the default mode can be seen in red, underlying here. And then we have 2 simulated covariates, one representing score on a testing battery, and the other representing the severity of some disease. And so to give you some idea of what the spatial layout of the data look like, we have the underlying default mode network in red. We have the covariate effect for score, where the location of that effect is going to be a green cube right here. Similarly, for the disease severity, the covariate effect is going to be in this blue cyan color. And then finally, we included an interaction between score and severity. That's going to be this yellow cube right here. So then we generated data from 3 visits. And for each visit, we slightly increased the intensity of the covariate effects. So now I'm going to shift over to actually showing the toolbox. And so I'm just going to show a few different sort of stages along the way of the analysis, and then we'll get into the visualization. So where we are right now, we're looking at the first panel of the toolbox. And I've gone ahead and I've set up an analysis folder, and I've loaded in the data. So for our toolbox, what we take is the data input is just a CSV file where the first column has all of the different paths to the Nifty files, and then the remaining columns have the covariate values. So once we've read in the data, we can do our model specification procedure. So if we click on select covariates, this will open up our model specification window. And so at the top of this window, we have the current model. So the first column is going to give you a list of which subject and visit you're looking at. For the simulation data, I named them as subject number and visit number. But if you have different names, whatever the file path for your Nifty files is what will be loaded in here. And then we can see the individual covariate values for score and severity. So the first thing that we can do with this model specification window is add and remove covariates from the model. So for example, if we didn't want to include severity, we could select it and remove it, and the design would update here at the top. Then I can add that back in. Similarly, if you have a massive CSV file, we have, and you'd only want to include a few of the covariates in the model, we have remove all buttons, which can be used to take everything out of the model. But I'm going to add both of those back in. So the second feature here is that we can change the type of the covariates from categorical to continuous and vice versa. So for this example, both of these are continuous covariates, but if we wanted to take severity, say, and make it a categorical variable, we can just select it here and click on make categorical, and the toolbox will take care of reference cell coding everything. If you want to change the reference category, you can come down here, select the variable, and then we have all the different options here, and you can select any of these to be the reference category. I'm going to go ahead and switch this back to being continuous. Then the last thing that we can do with this window is we can specify interactions for the model. So when I talked about the way the data were generated in the beginning, I said there was an interaction between score and severity. So we can create that here by selecting score and severity from the drop-down menus and just clicking on add interaction. And so that gives us our final model specification for the HCICA model. So then we can just hit save and continue. Now there's some pre-processing and initial guess steps that I want to just skip over. And so I'm going to jump straight to the EM algorithm. And since this is sort of in the middle of the whole pipeline, I've created a video for it. So from where we were a second ago to where we are now, all we've done is we've taken the data and we've done some dimensionality reduction via PCA. And then we've generated some initial values for the EM algorithm using a temporal concatenation group ICA approach. So once we do those two things, we're ready to actually run the EM algorithm and estimate the model parameters. So we can do that by going to the second tab, which is called Run Analysis. Give that just a second. And so once we get to this tab, there's a few things that we can change here. So the first is the maximum number of iterations, and that's just going to be how long the EM is going to run for. And then we have two convergence parameters, so the global parameter and the local parameter. And the local parameter is going to be corresponding to everything that's voxel specific. So this is going to be things like the covariate effects. And then the global parameter is going to correspond to the rest of the parameters in the model. So things like the error variance terms. And so that generally will hit a steady point pretty easily. And so once you start the algorithm running, there are plots for the change from iteration to iteration in the global parameter and the local parameter. Generally, most of that is going to happen towards the beginning, towards the first few EM iterations. And so this data set's pretty fast to run, but if you're using a much larger data set, if you're watching these change plots and you feel like it's probably already converged, you can go ahead and hit the stop button at the bottom there, and it will terminate early and give you your results. So I'm going to give this just a second to finish running. And then once it finishes running, the hint toolbox will carry out the variance estimates, variance estimation using the HCICA model here. And that's generally going to be pretty fast, but if you have a very large data set, it might take a few minutes. So then once that is over with, we can move on to actually doing the visualization here. And so there's 4 types of things that we can visualize with this toolbox. The first is the population display maps. And so this is the average map across the average brain networks across all of the subjects. Then we have the subpopulation display maps. And this is going to let us specify specific combinations of clinical or demographic covariates and see what the model-based estimate for the brain network would be. After that, we have subject-specific display maps, which just let us look at the individual subjects' brain networks. And then finally, we have the beta coefficients display maps, which is where we can look at the estimates for the clinical and demographic covariates and where we can do hypothesis testing for those. So first, I'm going to look at the population display maps. So I'm just going to open these up. And the first thing I'll want to do is load an anatomical image. You can just come into File and do that. So that's loading the anatomical. So now there's a few things in the viewer window. There's a few different panels. So the first at the top here is just showing the 3 different slices of the brain. You can click around here and the other 2 will update as needed. And then this bottom left panel here gives information about the location of the crosshair. So this can be used if you find certain regions of interest and you want to look up their anatomical meaning later. It can be used for that. And then in the middle here, we have IC selection, so we can decide which brain network we want to look at. And then we also have masking and different levels of thresholding by z-scores or raw values. And I'll talk a little bit more about that in a second. And then our third window here is going to let us select which visit we want to view. So right now we're viewing the aggregate map for the first visit, but if we want to add the aggregate maps for visits 2 and 3, we can just click here, and the toolbox will open up the viewer for those other 2 visits. And so these are all linked together. So if we click somewhere on one visit, the crosshair will follow on the other 2 maps. So one thing that we might be interested in here is seeing trajectories to change over time in the voxels. And so we can do that for the longitudinal data using the View Voxel Trajectories drop-down menu. So if we select this, this will open up a trajectory viewer for us, and we can see for the place in the brain that we've selected, we can see what the intensity of that voxel is at visit 1, 2, and 3. And so if we click around in different regions of the brain, we can see how that's changing. Another thing you can do here, if you're interested in creating something like a spaghetti plot for several different regions of interest, you can use this Save Current Voxel button. And so if I click around and just save the voxel that I've selected, you can see that those are stored here in blue. And so you can get some idea of the trajectory over several different regions that way. So I'm just going to close that out. And so the other thing that you can do here is threshold and create masks. So the first thing I'm going to do is convert these to z-scores by pressing this button here. And now I can drag this threshold slider and threshold the results from the HCICA analysis. So I'm going to just manually set this to 1.64, and you can see the voxels that survive that thresholding. And so we can create a mask based on this by clicking this Create Mask button. And there's 3 different types of masks that we can create. So there's the single visit mask, which is going to be based on a single selected visit. So whatever it outputs will just be the voxels that survive thresholding there. Then we can create a union mask, which will take voxels that survive thresholding in any of the different visits. And then finally, there's the intersection mask, which will consist of voxels that survive thresholding across all of the visits. So I'm just going to go ahead and create an intersection mask here, and save mask. And so now I can select this from the drop-down menu. You can see that the only voxels remaining are those that survive thresholding across all of the different settings. So that's a bird's eye view of what the population aggregate window can do. So now I'm going to switch over to the subpopulation window. And so now there's nothing here yet because we haven't specified any subpopulations, but this is going to add this control window at the bottom right here, where we can add subpopulations and we can specify their covariate values. So I've added a first subpopulation, and I'll say, giving them a score of 1 and a severity of 1. And then for the subpopulations, your interaction terms will automatically update. And so then we can view, say, the first visit for this subject or this subpopulation for the second independent component. Then we can add a second subpopulation, so maybe a score of 1 and a severity of 2. And then that will add it to the viewer selection window, and we can then look at it like this. And so what we can see here is our first subpopulation is on the first row, and our second is on the second row. And you can see these squares coming through here, which is what we would expect to see based on the way the data were generated. So that severity covariate effect and that interaction are coming through more strongly here. And then finally, we can apply the mask that we created a second ago to restrict these just to the areas that survived that thresholding based on the aggregate maps. So then finally, I'm going to switch over to the covariate effect viewer. So this is going to be the window that you would use to do hypothesis testing based on the HCICA model. So your different covariate effects will appear in the viewer selection here. So right now, score is being viewed, but I can also add severity, and I'll go ahead and add the interaction as well. And then we can convert these to z-scores using this button, just like before. Now, these z-scores are going to be based on the HCICA model-based standard error estimates. And then we can do some thresholding to do hypothesis testing. So I'll use a cutoff of 2.68. And we can see that the things that survive the thresholding are going to be these cubes here, which correspond to what we would hope to see based on the way we generated the data. So that's viewing just regular effects. We can also do hypothesis testing for contrasts. So if I come down here and I select the contrast view, this will again give us a covariate contrast place where we can fill out the different contrasts of interest. So I can press on add new contrast, and we can see, for example, for a 1 unit change in score, no change in severity, we can see what the corresponding contrast would be. And because I've already got the z-scores checked here, it's already converting them to z-scores and thresholding based on this cutoff here. So these would be the voxels that would survive thresholding here. And we can see that it's those 2 cubes, just like we would hope based on the way the data were generated. We can do another example, say no change in score, a -1 unit change in severity, a -1 unit change in this interaction. We can view that as well. We can see this time the negative effects coming through. So those are some of the main, functionalities of this hint toolbox. And then I'll just pull up some references here. So here's some of the references for some of the different HCICA approaches that have been developed, as well as the main reference for this toolbox. And that is it. And I'd be happy to answer any questions. Thank you.
So for next, our fourth speaker is Dr. Hernando Ombao. So he is professor of statistics under the Division of Computer Electrical Mathematical Science and Engineering at KAUST. So Professor Ombao's research interest is in the statistical modeling of time series data and visualization of high-dimensional signals and images. The talk he will give to us today is Toolbox for exploring interactions in multivariate time series. Let's welcome him.
Thank you very much, Shijia. So I guess I can now share my screen, right?
Yes, you can share your screen now. I think you're the co-host. Thank you. Okay,
great. So everybody sees the PDF at this point. Okay. So I have a talk on Wednesday, which is actually more of the methodological version of this talk today. Today I'll focus a bit more on the toolbox, and this is joint work with my collaborator Marco Pinto, who is in Oslo, and he really takes the credit for developing this software. So my goal today is to present this software. And the idea behind the software is for us to be able to explore some interactions and dependence in multivariate time series, and in particular in brain signals. Okay. So this is our starting point, is that we have an EEG, electroencephalogram data set with multiple channels. So here I only show 6 channels for now. And the goal is to be able to uncover some dependency between these different channels. And there's not one unique way to characterize dependence. And our goal in actually this project is to look at a spectral approach to characterizing dependence in a multivariate time series. So this is the basic idea. You think of this as a brain kind of shown sideways. So you have your latent sources on the cortical surface, which we do not directly observe. But what we observe are these data collected by the sensors. So these are the different channels. And the goal for us is to develop dependency between these different channels. We don't do source localization, but rather we directly model dependence in the sensor space. So take a look at X1 and X2 here. These are the observed time series. And one way to model dependence is to look at correlation, for example, between these 2 time series, X1 and X2. So you can model lag 0 correlation, lag plus or minus 1, and so on by fixing one time series and then shifting the other forward, backward, 1 unit, 2 units, and so on and so forth. Okay. However, the time domain dependence measure of correlation is global in the sense that I can actually decompose the 2 time series into their different frequency oscillations. So here I use Fourier, for example. However, one can use other types of decompositions. For example, wave decomposition is one possibility. But here let's focus on Fourier. And so we decompose X1 and X2 in terms of their low frequency, that's the delta band. Middle frequency, theta, alpha, beta. Okay, so these are, delta is 0 to 4 hertz, theta is 4 to 8 hertz, 4 to 8 cycles per second. Alpha is 8 to 12, beta is 12 to 30, depending on whatever convention you're using, but approximately that range. And gamma is 30 hertz and above. And so for us, we can model dependence between X1 and X2 through these different frequency oscillatory activity. So we take a look at the dependency between the delta component in one time series and the delta component in another. And then based on that, then we can develop a number of different dependency measures. So take a look at this figure. So at the top, we have the observed EEG signal. And then we decompose that in terms of the theta, beta, and gamma, for example. Now, if we take a look at the correlation as our measure of dependence between the F3 channel, so that's in the frontal lobe, and the occipital channel O2. By the way, O2 should be on the right side. It's on the right side of the brain. So you can take a look at correlation between these two, lag zero correlation, and then we compute correlation to be 0.547. So now the next question for us is, what is the frequency oscillation, in fact, that drives this dependence? And so we take a look at coherence, let's say, or correlation in terms of their theta waves. So we decompose the F3 channel and the O2 channel in terms of their different frequency oscillations. And if you focus on the theta oscillation, then we can compute the correlation between them, lag zero for the moment, as equal to 0.541. Now you can also compute the correlation between the beta waves for F3 and O2, and then you get 0.122, and so on and so forth. And so here, correlation on top, this is in the time domain. The correlation between F3 and O2 is 0.547. But if you break this down into the different frequency oscillation, then we in fact obtain different values of dependence. So at the delta component, it's 0.284. At the theta component, it's 0.541, and so on and so forth. So of course, dependency can go beyond just linear dependency. So for now, I just computed the correlation between these different oscillatory activity. But later on, one in fact can and should look into other nonlinear interactions between them. So for example, I can take a look at dependency as a function, dependency between different between these different oscillations as functionals of them. So dependency between the absolute value and other types of functions. So in this toolbox, we will present a number of different measures of dependence that is already available in the literature, and some of them we have tried to write papers that's a bit more pedagogical. So we put a framework under which all of these different dependency measures, in fact, fall under. Okay. So this is, we'll take a look at coherence, partial coherence, partial directed coherence, and also, as time permits, lagged dual frequency autocoherence. So here, we'll take a look at dependency between different frequency components, for the moment only within a single channel. Okay, but later on, we can also take a look at dependency between different oscillations, let's say the alpha band and the delta band at the different channels. So this is a software architecture that's developed by my collaborator Marco Pinto. So it runs out of the R Shiny, and I'll go straight now into the demo. So I'll stop sharing the PDF, and I'll now share the toolbox itself. Okay. So here we load the different. You can load different data sets via the RStudio command window. And from there, this graphical user interface actually is initiated. And so on the left panel, you see a number of different tools. So you can take a look, you can model correlation, you can explore correlation as a measure of dependence, coherence, partial coherence, PDC, partial directed coherence, lagged dual frequency autocoherence. And then we also have some tools here for reducing dimensionality in the multivariate time series. Okay, so I won't go into that today, but these two are also in fact discussed in the paper on which my talk on Wednesday is based. So let's start with correlation. So if you're ready, this is familiar to everybody. So these are the different time series, the input. And then on the right, so here you'll see this correlation, pairwise cross correlation matrix. And then we have the graph. Of course, thicker lines denote stronger magnitude of correlation. So let's go now to coherence. So with coherence, So with coherence, we can, okay, so at the very top, you see the spectrum estimation parameters. So there are a number of ways to estimate the spectrum, because once the spectrum is estimated, then we already obtain estimates for coherence. So that's one way to think about it. Another way is to actually do the actual filtering, like what I have demonstrated. So the framework is that the filtering idea holds true here, but in terms of how you actually implement it, then there are a number of ways to do it. So one is here we estimate coherence by directly estimating the spectral matrix, and so we have there's a parametric estimator. That's an option, and for now we're using the vector autoregressive model as our parametric model. You can specify the lag order, 2, 3, and so on. Now, so if I stick for the moment with the parametric estimator, then we're fitting a VAR model of order 2. Now, there are a number of ways to, in fact, estimate the VAR parameters. And one of them is just by ordinary least squares estimator. The other is we can implement this Lassell approach. This was developed by Lichuan Hu et al., where in the presence of high dimensionality, their approach is to, first of all, fit a lasso on the VAR model. And then at that point, they identify the coefficients that were actually estimated to be non-zero, and then those are re-estimated via least squares. And then, of course, now there's here we implemented the big VAR approach by Matheson, Bian et al. At Cornell. So in terms of the parametric modeling of coherence, then we use VAR for the moment. Now there are also the non-parametric estimator, and so here this is just when we say naive smooth FFT, this is really the smoothing the periodograms. Okay? And we use the Gaussian window. You can specify the span of the frequency window. I must add here too that there are already a number of automatic ways of estimating of selecting the span for periodogram smoothing. So one of them was by Thomas Lee, which is based on least squares. And the other one, which I actually developed some 20 years ago, it's based on gamma generalized cross-validation, because periodograms, in fact, are asymptotically distributed as gamma or chi-squared random variables. So there are, but here we're not implementing the automatic selection of the smoothing span. We just let the user specify that. And now with a non-parametric estimator, the reason why we do this is because we will implement later on a dependency using partial coherence. And so we implemented the work by Fecas et al., where to estimate partial coherence, they first estimate the spectrum, and then the spectral estimate has to be inverted in order to obtain the partial coherence between different pairs at different frequencies. And now, in order for the spectral estimate, the spectral matrix estimate to be well conditioned, what Fecas did was, in fact, to do a weighted average of the non-parametric estimator, which is a mildly smooth periodogram, and a parametric estimator, typically a VAR, although he started out actually with some regularization based on the identity, based on the diagonal matrix. Okay, so here to estimate coherence, we can do the same. So in the end, our final estimator of the spectral matrix is some weighted average of a parametric estimator and a non-parametric estimator. Now, the weight actually was derived by Fecas et al. To be inversely proportional. So the weight for the parametric estimator is proportional to the mean squared error of the non-parametric one, and vice versa. So if the other estimator has a high mean squared error, then a high weight is put on to the other estimator, and vice versa. So these are how we specify the estimation of coherence. And so here now, in terms of the plot, suppose I just stick with the parametric estimator for now, of var of order of lag 4. This is using the ordinary least squares. Then we can look at different pairs of channels, so FP1 and F3. Then we can also, because in fact coherence is frequency dependent, so this is at frequency 3.95 hertz. So you can slide this to obtain the dependency at the different frequency bands. So this particular data was sampled at 200 hertz, and therefore the frequency range goes from 0 up to half of the sampling frequency. So if I take a look at, for example, here 19.53 hertz or about 20 hertz, then we see that the F4 and FP channels are very strongly coherent. Okay? And then, so we have this graphical network here as well. Okay, so let's go now to partial coherence. So as I was saying earlier, to estimate partial coherence, then the approach of choice for me is a semi-parametric one, as proposed by Mark Fecas. And as I said, that's a weighted average. And the interpretation of partial coherence is as follows. If I'm looking at the partial coherence between two channels at, let's say, some frequency band, the delta frequency band, then partial coherence looks at the dependency between those, but removing the other delta oscillations of the other channels. So let me pause for a second here because I see a question. Maybe that's for me. Okay, 4 minutes remaining. Thank you, Shijia. Okay, so next is so if we use a semi-parametric estimator with some mildly smooth periodogram, then these are the on the right here. This actually is a partial coherence matrix. Okay, instead of coherence, this is partial coherence. And then now we can take a look at different pairs of these and so on. Okay. Now with partial directed coherence, this actually is based on the measure developed by Bacala and Sameshima. So the ones that I have just described earlier, coherence, partial coherence, all of these do not have directionality. But very often, especially in brain science, we want to know whether the activity in one brain region or in one channel precedes the activity in another brain region or another channel. And so we want to be able to capture that lead-lag relationship or that directionality. And so one way to do that is by using the approach given by Bacala and Sameshima, which again is based on a vector autoregressive model. So I'm going to end the demo here, and on Wednesday I will cover a bit more of these different measures of dependence and the justification for them, the motivation behind them. So I can stop sharing now and entertain questions.
So for our last speaker, she is Dr. Marina Vannucci. Professor Vannucci is currently the Noah Harding Professor of Statistics at Rice University. She also holds an adjunct appointment with the Department of Biostatistics at the MD Anderson Cancer Center. Methodology developed by Dr. Vannucci have found applications in chemometrics, high-throughput genomics, neuroscience, and neuroimaging. The talk she will bring to us today is titled User-Friendly Matlab GUIs for Bayesian Multi-Subject Modeling for fMRI Data. Let's welcome her. Thank you.
All right, thanks. So I think you can hear me and then you can see my slides. So I'm going to just very briefly talk about two user-friendly Matlab GUIs that have been developed by a student in my research group. And actually, the student who was supposed to give the talk last year, and he would have given more of live presentations of the GUIs, but he has now graduated and he has a job. So I'm going to do my best to present the software. And then I have like a 5 minutes video at the end by the student, Eric Cook, who will illustrate some of the features of one of the two GUIs. So the first one is NP-based fMRI, and it implements a Bayesian framework for activation detection of for task-based fMRI data on multiple subjects. The approach is a Bayesian, and so the peculiarity of the approach is all about the way that we structure the prior. In particular, we have a prior, especially informed non-parametric variable selection prior that can account for between subjects heterogeneity. And I will give you a brief overview of the prior. It's in spite of being a Bayesian approach, the inference is relatively fast because we were able to essentially bypass the MCMC by implementing a variational base algorithm, so that works pretty well. The output is activation detection maps for at subject level as well as at group level. And as a bonus for the type of prior that we used, we actually get also a clustering of subjects that share similar brain responses. The second user-friendly Matlab GUI is BVAR Connect, which implements a Bayesian vector autoregressive model for the estimation of effective brain connectivity, so directed connectivity, based now on resting state fMRI and again on multiple subjects. And here we assume that we have groups of subjects. So instead of discovering clusters of subjects like in the previous GUI, here we kind of assume that we know groups of subjects. We might have a group of healthy patients and a group of disease patients. And again, the approach is we're going to estimate network connectivity networks, both at the level of the group and again at the level of the individual subject. The approach is Bayesian, and so again, the peculiarity here is in the prior, the way that we set the prior. In particular, we have again a variable selection prior that flexibly allows the integration of external information, for example, other types of imaging data like structural data. Again, the inference is done by a variational base, and so everything works pretty nicely and relatively fast. Okay, so starting from the first GUI, so the NP-based fMRI for the detection of activated brain regions. Again, I'm going to walk you through the model and in particular the prior structure, and then I have a few slides with snapshots from the GUI. The model is the classical GLM model that assumes that the fMRI data is given by the bold signal past the noise. We do model the noise pretty heavily. In particular, we allow for correlated noise, low frequency correlated noise, but I'm not going to spend time on this today. The bold signal is modeled as the usual convolution of a stimulus function plus the hemodynamic response function, and we allow a couple of different hemodynamic response functions in the GUI. The important aspect of the model with respect perhaps to other activation detection software out there is that we implement a Bayesian approach, and therefore we have kind of an elaborated prior, in particular on the regression coefficients of the model of the bold signal. So here in particular, the prior that we use is a spike and slab prior that allows for zero regression coefficients and therefore for the selection of the activated voxels. I think I have, yes, on the next slide, I have the form of the prior. So again, it's a spike and slab prior, so it's a mixture of two distributions, but one of the two distributions is a spike at 0, so that it allows, gives probability to the event beta no equals 0. And the prior allows for the modeling of correlation, both nearby correlation, local correlation, as well as distant correlation among voxels via imposing for the local correlation via imposing a Markov random field prior on the indicator of the mixture, this parameter gamma nu that has a Markov random field prior that you see here on the slide that has a couple of parameters. And so in particular, through this prior, the prior probability of a particular voxel to be activated will depend on whether the nearby voxels are activated or not. And we also incorporate a distant correlation among voxels by allowing for so-called non-parametric spike and slab constructions, where the slab portion of the spike and slab is actually non-parametric prior that imposes some clustering among activated voxels. So this prior in particular would achieve clustering of voxels within a subject, which are clustered according to similar activation patterns. And we exploited this prior, in particular one in the extension of the model to multiple subject, because in the case of multiple subject modeling, this prior allows to do clustering of voxels within subjects and across subjects, and therefore it allows to cluster subjects that have similar activation patterns. So this is kind of a depiction of this feature of the prior. So we can cluster subjects that share similar activation patterns, and then within individual subjects we can cluster voxels that share that are correlated, they share similar activation strengths. Okay, so I have a couple of snapshots of this GUI. So it's in another slide. I have the link to the GitHub, so you can get it from my GitHub at RISE. And once you install the GUI, it has a window or an interface of this type up here on the slide that has two different interfaces, one for model fitting and one for visualization. So if you click on model fitting, you can load the data and set the parameters and run the model, go away, and then come back, load the output, and view the output through the visualization interface. Here are a snapshot of some of the features of the model fitting interface. Again, you can set the output file where your output will go, set the number of subjects. We can do both 2D and 3D analysis, meaning we can analyze single slices at voxel level, or we can do a whole brain analysis where we parcellate the brain into regions of interest. So that's the 3D analysis. And then there is some parameter setting. For parameter setting, I think I have another one. Yeah, so for parameter setting, we have a full set of default values, but we also allow the user to specify your own hyperparameter. So if you understand the prior well enough to be able to play with the different parameters, you can actually do what we call sensitivity analysis. If not, there are a set of default priors that we have been tested quite extensively, particularly on simulated data. And then, as I said, the second interface is a visualization interface. So here, once you load the results from this file results.mat, then you can visualize the activation maps, essentially, as I was saying, by subject, but also by cluster, so by groups of subjects that have been found to share similar activation patterns. And you can also view contrast maps by subject. I think I have one slide here with the GUI to plot the activation map by subject. It's a little bit similar to what Joshua was doing, in a sense that you can select different views of the brain, different slices, different views, and different colors, and so on. So it's pretty cool to play with it. So you get the activation map and what we call the probability map, which is the posterior probability of activation for the individual voxel. So this is the map that that gets thresholded to plot the activation map, and you can select your own threshold or calculate essentially Bayesian FDR threshold. Okay, and then you can also, as I was saying, plot activation maps by cluster. So you can see actually the cluster, you can break the subjects in different clusters according to this dendrogram, and then plot activation maps by cluster. Okay, so this is the first GUI, and this is the link to the GitHub. There is a accompanying paper that describes the GUI in details, and this paper here published in Statistics in Bioscience in 2019. And Eric Cook, as I was saying, is the student who implemented everything. The second user-friendly GUI that I want to present is BVAR Connect, and this is a GUI for effective connectivity, so what I would call directed connectivity based on fMRI data on resting state subjects. Here, the idea is to have subjects, possibly groups, different groups of subjects, and the approach is based on vector autoregressive models. We have heard about vector autoregressive models in several different talks today. So given the bold data, in this case, we are assuming parcellated data. So we're only working on 3D parcellated data in a set of regions of interest. And so given the parcellated bold data for subject S in, say, group G, so we model the data through an autoregressive vector autoregressive process of order L. And then we focus on, in our modeling, we focus on the set of coefficients of this VAR process, which are those are the subject level, lag specific, effective connectivities between regions for the particular subject. We impose a prior on these coefficients, which is essentially centered on group level effective connectivities, and then we impose a spike and slab construction on the group level effective connectivities. So we select non-zero effective connectivity at group level and infer the subject level group connectivity as a deviation from the group level. Okay? So I know that I'm going perhaps a little bit fast, but there are papers with these formulations. So this is the spike and slab prior at the group level. It's a little bit more complicated than before because being on the entire brain, we kind of allow some smoothing across different regions, but with the software, you can also turn this feature off. And then a peculiarity here of the approach is that through the prior on the indicator of the spike and slab, we can allow for the incorporation of additional covariates. For example, we used MRI data, so information on structural connectivity, so that the prior probability of there be a connectivity between two regions is higher if there is evidence of structural connectivity between these two regions. But the construction is flexible, and you can think about informing the prior in different ways. The software is also quite flexible. So this is one snapshot on BVAR Connect, which is again in my GitHub. It has very similar to the previous GUI. It has two interfaces, one for model fitting and one for visualization, which is now through connectograms. And we also allow to export the data in a CSV format, because once we started building connectograms, we realized that there is so many different ways to plot these connectograms, and there is so much software available out there that it's probably easier for our users to extract the output, extract the connectivities, and then choose whatever software they want to plot the connectograms. But the software, the GUI, does have some ways of plotting connectograms. And now at this point, if I have 5 minutes, which I think I do, I would like to plot to just show you a video that Eric has prepared on BVAR Connect. Before I do that, so that I don't have to come back to the slides, I want to thank, again, Eric Cook is the student who implemented the two GUIs, but Lin Zhang and Sharon Chang are the students who first worked on the activation detection models and the effective connectivity models. And the work is all joint with Michele Guindani, as you see hereby. So let me see if I can quickly start. It's only 5 minutes, which I should be able to have. Okay. There.
Hello, everyone. In this video, I will provide a brief walkthrough on how to perform model fitting and visualization using the BVAR Connect software. The files and resources to install the GUI can be found on Dr. Vannucci's GitHub webpage within the BVAR Connect folder. Upon starting the BVAR Connect software, the user will be prompted with three main options, which are model fitting, export data to CSV, and connectograms. Model fitting is the option that is used to implement the variational model within the user-specified data and parameters, and the later to provide means to inspect the output from the model fitting procedure. In this video, I will briefly go through a subset of features available within model fitting and connectograms. So let's first start with model fitting. For your information, this exercise can be replicated by using the real data med and demo script files, which can be found in this example data folder. So once we click on the model fitting ribbon, we are prompted to specify 5 sets of inputs in a chronological order. The first input is the output directory, which is a directory where the model output will be stored in the MAD format. Then we have the fMRI data, which is a MAD file that should contain a 3D array storing the fMRI time series data for different subjects across different regions, a cell object storing the name of each ROI, and variables storing the number of legs for the AR model, the number of groups, and a variable which stores group membership information for each subject. Next, we have structural data, which is a MAD file containing structural information across the regions of interest. This is an optional step and can be omitted if the user does not have structural information. So now we're done with the data part and we proceed to the prior setting. In case of the prior specification, the model is defined with default parameter settings which the user can modify. Once we have gone through the list of inputs, the user can confirm that everything has been set correctly by looking at the input description and then run the model by clicking on the Run Model button. Once the model has run successfully, you will find a MAD file named out in your previously specified output directory. We can now use this output for visualization purposes. As a disclaimer, this visualization option is only applicable for a lag 1 model. Okay, so let's explore the ROI-based connectogram. So this is a feature used to visualize effective connectivities among different ROIs. So once we click on the ROI-based connectogram, you will be prompted to load the output from the model fitting procedure. So once we click on the Define Connectogram button here, we first need to specify the group we're interested in reviewing and set the corresponding option to active. Then we specify the options for the remaining groups and confirm the specification. So the choice of options are used to decide whether we want to look at connectivities that are present across all groups, subset of groups, and so forth. For more details regarding how we define the connectogram, please refer to the README Markdown file on GitHub. So upon confirming the connectogram, BVAR would extract the necessary information from the model output and display how many regions have connectivities, which in this case, the 82 regions out of 90 regions. So now that we have defined the contrast, we can visualize our results by clicking on the Plot Connectogram button. So a cool feature with this connectogram is that the figure is interactive. For example, if the user is interested looking at a subset of specific ROIs, one can click on the node to remove all connectivities that are branching from the specific ROI. And afterwards, by clicking on the subset ROI button, which is found on the bottom left, you can remove the ROI names from the graph as well. Then by clicking on the show all button, you can recover the original image as before. Aside from this, the user can also specify the ordering of the ROIs by either inputting a vector yourself or inserting it from a table as the following. Then once we confirm the order, you will find that the ordering has changed as desired. This concludes the walkthrough of BVAR Connect. I hope you found this video to be helpful, and I thank you for your attention.
All right, and that's all I have. The video is also in GitHub, as well as the company paper for the software. Thank you.